Welcome to Interview with a Biologist. I'm your host, Marie Locito, and we're going to be joined today by a number of special guests. The students in the spring 2026 cell biology class completed independent experiments that they created after looking at changes to human cell cytoskeletons after exposure to various treatments such as microplastics or caffeine. Today, we'll be hearing from different students to learn about their results. First off, I'm joined by Evan Schulz, a TA in the lab, who will be telling us a little bit about the cell biology course and the basic principles behind this experiment. Evan, as a graduate student at KU, what do you enjoy most about teaching undergraduate classes?
I really enjoy seeing students grow and learn and also get excited about the material or whatever it is that we're doing. Especially this class really is an epitome of that. Students grow so much from the beginning to the end because they're really doing what they're learning is really hands-on. So instead of just shoving a bunch of information in and then regurgitating it, they're actually learning and growing as scientists, which is so much fun. That is really fun for me, yeah.
Yeah. I mean, I have a very, very limited amount of student interaction, especially with classwork, but I do a student in the lab research, and it is so satisfying to sort of hear about the growth from where they started to where they end up when I'm talking with them. So as a TA, having more hands-on, I bet that just feels so good.
And it's so much fun because students they figure out kind of what excites them about biology while they're in the class. And that's so fascinating for me, is to also learn about what they're interested in and see that kind of develop.
You reached out to me about this whole thing and getting it put together. What sort of made you decide to think like this will be a great thing for the podcast?
Well, there's kind of like two reasons. The first one is that more than any other course I've TA'd for here at KU, I really wanted to share about this course because I wish I could have taken this course as undergrad. This course, Dr. Jennifer Houston, our lab director, has really built a course that has a lot of real value, not just for future scientists, but future doctors and future medical professionals. You really get hands-on training in being a biologist and developing real scientific thinking and conducting actual biomedical experiments. You're not just learning off of a textbook. You're actually doing these things, which is, as somebody who has transitioned in the past from being an undergrad to trying to work in real-life labs, that's a huge deal. That is a huge challenge, and getting ahead of that is like something I wish that I had. These skills are the actual skills that get you a job as a scientist or as a biomedical professional. And more, whether it's grad schools, medical schools, they're all looking for applicants who understand these skills, can actually perform these skills, and think critically and scientifically. And there's no way that you can learn any of this from just a textbook or online or asking AI. This is something you have to actually physically do. And I think that you guys are exceptionally well qualified coming out of this course for doing a lot of these techniques. I was blown away by how much you guys have grown and what you guys have done. And that's sort of like the second reason, is that I'm actually just really, really excited to share about this because I'm really excited and really proud of your guys' work and your experiments. You guys designed truly original and fascinating questions and figured out how to test them, and you got really beautiful data from the experiments you guys ran. So that was just a joy.
I started at KU coming up on 5 years, so officially 4 years ago, and seeing how the class-based research has just developed since I first started, when we had maybe half of a cure class, to where it's at now has been really inspiring to know that there are other opportunities for students to get these kind of hands-on skills and learning even outside of, admittedly, a limited number of research spots that are just available. So it's exciting to kind of have all these students in to talk to us and learn more about how this has impacted them.
Mm-hmm. And I actually know that from the perspective of a lot of people looking for undergrad help, they're looking for whether or not they've done courses like this, because they'll have already done and learned basically all the key critical components of how to work and function in a lab. It's a huge boost to anybody's resume or application.
Could you tell us about the experiments and techniques that this lab covers and offer to the students?
Yeah, so There are 4 key ones that they learn. The first, and honestly one of the trickiest, but also one of the most meaningful, is cell culture. So they spend the whole semester culturing cancer cells, and this is a technique that is foundational to all biomedical research. We've all heard of the huge impact that just the single HeLa cell line has had on medical history. And all research going forward uses models like this, and cell culture is a critical technique. They also learn Western blotting, they learn immunocytochemistry, and they learn fluorescent microscopy. For me personally, as a cell biologist, I consider these techniques to be the core techniques for any researcher, whether they're a doctor or a PhD. They are using these core techniques to investigate their questions in the field of biomedical science.
So would you say that students in any sort of field of biology research or STEM research in general could benefit from this class?
Absolutely. I think not just people focused purely on STEM, but also medicine. I think that for people in medicine, understanding these techniques is, as I kind of said before, really critical, because even now, a lot of our medicine is based off of these techniques. Every single chemotherapy drug has had research done on it using these techniques. And every single drug has also had that same rigorous process going through the pipeline of development. So understanding how these experiments work, what this data looks like, and how it's interpreted is critical for doctors when they're learning about new innovations in the field of medicine and reviewing old ones. For anybody in the STEM field, this class, more than any other, really teaches you critical scientific thinking that's broadly generalizable. So these students came up with their own questions from their own curiosity, and they went through a really rigorous process of figuring out how do we actually test this in a scientific way and ensure that we get usable results. That's huge, and I think it's something that anybody in any STEM field really needs to master in order to be a successful scientist. The part of the lab that I enjoy most is definitely their independent projects. I think it is just so much fun to see them be creative, and I really believe that this is like an amazing form of creativity, is coming up with these questions, asking these questions, and then testing them in a scientific way. There's just not just seeing them grow as scientists, but also just their actual results that they got were fascinating. There was a group that did an experiment with microplastics, and not just seeing what that did to the cells, but also, so they boiled a tea bag, and then they extracted the microplastics that came out of that tea bag, and it was actually visible to the human eye. Loose leaf is the way to go. Yes. Get yourself an aluminum strainer, friends. Yeah, whatever that little doohickey is called, I still need to get one. I remember after I saw those results, I tried to make a cup of tea by just cutting the tea bag and pouring it into the thing, and it was a mess. It just blew me away that you could actually visibly see the microplastic pellet from that extract from a single tea bag.
I'm deeply upset, and I'm going to be thinking about this. So, fascinating. And then, is there anything we haven't talked about that you think we should?
That's a good question. I think I really encourage anybody who's interested in taking this course to reach out to Dr. Jennifer Houston. It is a really fun course. I would say that you don't have to worry so much about memorization. You really develop and grow as a critical thinker, and every lab that you do, I personally think, is really fun and also really valuable. So if you're looking for a course that really is going to improve your resume and yourself as a scientist, this is the one that I would have taken.
I'm joined today by Ariana Siddique. Ariana, would you tell us a little bit about yourself?
Hi, my name is Ariana, and I'm a senior studying molecular, cellular, and developmental biology with a minor in Spanish.
What is sort of your end goal for once you graduate?
I start my master's in public health program this summer, and then I'll do that for a year and hopefully go to medical school after.
Is there like a particular kind of doctor you're looking into being?
I think I'd be happy to do like any field of medicine. I kind of love all of it.
Yeah. I mean, I guess that's kind of what rotation is for, is to get a feel for like, this is what I actually want to do and not just what I think. So very cool. Well, I'm excited to hear about where you are like 5 years down the line. So for this class specifically, would you tell us a little bit about your research project and why you picked that treatment?
So my partner Chelsea and I researched how different types of water would affect the actin cytoskeleton of HeLa cells, which are cervical cancer cells from Henrietta Lacks. And I kind of got the idea because I have fish tanks, and in those tanks you have to control the nitrate levels, pH levels, temperature, and all sorts of other chemicals. And so since we use water as a dilution for most of the solutions in our lab, I was curious how that would have a maybe confounding effect on our experiments. And so we tested 3 different types of water, all at the same temperature, and observed how they affected the structural integrity of the cells.
Could you give us an idea of what those 3 different kinds were?
So we did kind of a gradient of hypotonicity. So we did molecular biology grade water, which is purchased from like a science factory or something like that. It just appears in our lab, and it's like the most pure form that I could find. We use DI water, deionized water from a reverse osmosis filter that goes right into our lab in Hayworth. And we use tap water from Hayworth. And I took a picture of the lime scale that was kind of built up on the faucet. And we did some research and saw that it might be because of iron or different types of dissolved minerals. So we're kind of curious how that would cause an effect on the cells.
And just to butt in here, it looked like a cave formation. Like the
like
actual like faucet, like a stalagmite or mite.
Yeah,
it was
whichever one.
Yeah, it was kind of interesting to think about. Like, yeah.
It was nasty.
I think about like all of the cheap apartments I've lived in over the course of my life, and like just the mineral buildup that you'll get on shower heads. Yeah. And after Hayworth being built in the 60s/80s, depending on the side you're in, I imagine that's a lot of buildup. So I wouldn't be surprised you got some like drip. And part of me is a little terrified to know what the tap water did to these cells. So what were your findings?
So it was pretty cool because all of the different water had a really distinct effect. Like you could tell which one was which. Mm-hmm. And we did a control using PBS. Mm-hmm. I forgot what it stands for. But it's isotonic, so it should be like neutral to the cells, which kind of gave us an idea of what a normal cell would look like, and kind of went off of like past labs to remember what those cells looked like. And overall, most of the cells were damaged and kind of mangled, like the filopodia that are kind of sticking out from the cells had collapsed, and there were signs that it had lost adherence to the slide, which is kind of what HeLa cells are known for, being adherent. And I think the tap water was pretty menacing. Like, it was really kind of almost like hazy and collapsing, I guess is the best word for it.
Yeah. As compared to your
control,
PBS control. The PBS ones looked so healthy and really branched out, being like very healthy cancer cells. And then in your water one, like all of them were just very disrupted. Their actin structure was very collapsed. The cells were in the process of dying. Yeah.
Yeah. And we looked around the slide because there's quite a few cells on the slide, and it was pretty representative of what we were seeing. And the DI water we anticipated to probably be the least damaging, but it was still pretty severe. We were pretty surprised because DI water is what we use to make our buffers. And so if the DI water was causing so much damage, we were kind of curious, like, when we do different experiments to further disrupt the skeletal integrity, like how much of that is maybe because of just the water we were using.
And your exposure time wasn't very long, right?
We just did 3 minutes.
Oh, wow.
Because we were worried that it would just shock the cells, because any type of water that's not, like, buffered is going to destroy them, I think. Mm-hmm. And we were also really concerned about temperature because those cells are so sensitive to different types of shocks. Mm-hmm. And another crazy thing was that the molecular biology grade water, like, since it's so pure, there's nothing to buffer anything, like, those cells were by far the worst. So much just collapse. You couldn't see any filopodia, and parts of the cell were kind of missing, kind of suggesting maybe it had folded in on itself or just kind of maybe disintegrated, if that's possible.
Yeah. And so a lot of people think that, like, pure water is probably good to drink, but your results really showed that, like, in reality, the water that our bodies needs to have all these other ions in it. It needs to be balanced. The pH needs to be right. Otherwise, pure water, you don't want to just drink pure water from a laboratory because it actually will cause a lot of probably issues with whatever tissue it comes in contact with.
Right.
But yeah, your results were beautiful. You had beautiful staining. And one thing I didn't realize is just how, so you're going into a master's in public health, right? Mm-hmm. Your project is a really interesting kind of public health perspective because you tested water that comes out of our faucets in Hayworth, which is really interesting. And we all know Hayworth is a very interesting ecosystem. So, I thought that was really cool and had real world relevance.
Thank you.
Yeah, no, that honestly fascinating project. And I never really thought about the effect of water on cancer cells. They just, in my brain as a non-science person, they exist within your body and they're doing bad things. And, something just so ordinary as water can have an impact, have an effect on that is kind of astounding. And I guess this may be so far beyond what you know about, but generally when you have cancer, you want to kill those cells. So by seeing cell death within your line, I don't know, did that have any sort of correlation or relevance or make you think of anything as you were sort of unveiling these results?
I mean, I guess my thinking was that if it's so harmful to the cancer cells, which I think are, in my mind, more resilient than other cells, that how, the line of thinking for the research project was, if they're this bad for the HeLa cells that are so strong, what would they do to, a normal healthy cell that maybe isn't as resilient? And so I guess, my mind goes to drug delivery maybe, but if it's so harmful, again, it would probably also be just as bad as, the other adjacent cells that are healthy.
And definitely these cells can be used as sort of a model to get an understanding at the cellular level how different treatments might affect not just cancer cells, but cells in other places in the body. And one thing I think is really interesting is that it reminds, in the course, you guys also did work with chemotherapy drugs on the same HeLa cells. And that not only reflects what it does to cancer cells, but really importantly also reflects the side effects that come from those chemotherapy agents that have really nasty side effects throughout the body because those drugs, especially because they're not super targeted, they're going to be affecting all the other cells in the body as well. And so both your water experiment and the other experiments you guys did in the course kind of reflect that the model that we use in these cancer cells are applicable both to cancer and normal body cells. Yeah.
Right. And I think just to put into perspective, we did a 3-minute incubation with the different waters, but everything else that we did to do the actual permeabilization and staining to stain the actin cytoskeleton was also in the range of 2-3 minute incubations. And so time was very important to us, but we had an evacuation. And so
was it that fire drill day?
Yeah.
Yes.
Was it a drill?
Probably not a drill. There was something, as always, but
well, we were in our paraformaldehyde fixation part of it after we had already done the water incubation, so we don't think it had any effects on the experiment. But it was a little terrifying because time was they're just so fragile in that sense.
Yeah, you guys got lucky. Unlucky, but also lucky because you guys did fix before something happened. Mm-hmm. And then we all had to leave. It literally happened just at the right, the worst possible time because a lot of people were just in the middle of their experiments. So that was pretty crazy. But I think everybody actually managed to salvage what they were working on and people got really cool results.
Yeah, I mean, I'm glad it was salvageable and you didn't have to run the experiment again. What did you enjoy about doing this experiment?
I think my favorite part was getting to do the stain and kind of wait in anticipation of what we would see. I think we were expecting some dramatic results, but could not have anticipated just how dramatic they were. And I think just seeing something that's so deceptively simple, water from a faucet having such a detrimental effect was an exciting prospect.
So for this class overall, how do you think all of these experiments that you did is going to benefit you in the future?
I think it gave me a lot of critical thinking skills and problem solving, more so than like I would in the classroom, I think, just because like you run into problems that you would never think about. Like one thing we talked about when we were coming up with the project was visualizing the project, and then how when we visualized it, we saw like what made sense on paper didn't really make sense in practice. When we were actually doing the experiment, we ran into all sorts of issues, like for everything from an evacuation to I think we forgot to cover the light sensitive stain with a bucket to prevent light from seeping in, and that could have been detrimental too. But I think just learning how to bounce back, and I think salvage is a good word. Like I think in real life a lot of things don't go as planned, and you have to like think back and salvage it, and I think that class helped me like in a really practical sense.
Yeah, in terms of like any kind of research, troubleshooting is something that happens at every stage. You guys really did a great job troubleshooting your project in real time, and like if it can go wrong, it will go wrong, and that was so clear with your entire class because the fire alarm went off, and that just you guys had to troubleshoot how could this have affected our experiment, and you had to use your understanding of the techniques, which you guys demonstrated very well about how this might actually is this going to negatively affect the results? Is this going to make it so that the results are usable? Are they not usable? Does it provide or cause any limitations to what we see afterward? And you guys did a great job.
And one thing Evan said like throughout the class was to think like curiously and critically about everything like in every day. And I feel like our class had a lot of projects that kind of demonstrated that. So I think 2 different groups did sunscreen and how that affects the cells and things like that. So I think that's something I'll take into the future, is to just think like a little deeper and ask like why things are the way they are.
Yeah, I mean, honestly, a great skill set to take from, what you just said. And then also when you had mentioned like the problem solving around problems arising, like between work and just real life, that is something so good to sort of instill now and just always be kind of mindful and thinking.
And I just want to give a shout out to Dan, who runs the greenhouses in Hayworth. We used his sunlight for the sunscreen experiments. And everybody in your guys' class came up with really, truly original, creative questions and experiments to run. And it was really, really fun to see you guys do that.
Yeah, and I guess if you happen to know, sunscreen in the greenhouse, yes, no, is it even more necessary than outside of it?
So they actually exposed their cells sort of like outside the greenhouses, but on that sort of
the patio
veranda. Yeah. And so the interesting part though was the cells were in polystyrene plastic, so the UV that was getting through was mostly UVA, which is something. So essentially, the results, they actually did see that if you don't use sunscreen, you do get damage to the cells, even just through UVA coming through plastic, which is relevant for us in terms of glass window panes or other ways that sun can get into interior environments. Which is kind of freaky to think about. And but their results are also really, really cool. They tested mineral versus chemical, and it was a very interesting experiment.
Yeah, sounds like it. And sorry for getting off subject, but like as someone who's like, I got a sunscreen, I had to know.
And unfortunately, they said that mineral was better, which is unfortunate because that's the one that is so annoying to put on.
It's true. And then like I always have problems where like after I've been wearing it for a while, it starts to like gum
like
up a bit. Thank you, pill.
Yeah.
And I'm just like, but I want to look smooth. So, yeah, good to know. Okay, and then final question. What part of the lab did you enjoy most?
I think my favorite part, I was really excited when I saw we were learning about HeLa cells because I read the book about Henrietta Lacks, and she was always something that I was like, oh, this is, terrible what happened to her, but such an interesting story. And I was really excited because one of the first things we did in class was we learned about the cells, but we also talked about like the public health perspective of it too, which was like very exciting for me. And it was really cool because it kind of showed me that like there is a place for both like public health research and medicine that kind of coexist. And it was just exciting to see that in practice because I feel like when you're in college, you're like, oh, I would love to be in the intersection of all of this, but then you get to actually do it. And that was really cool.
Yeah, it's great in a way to have that hands-on sort of experience prior to entering the workforce and being able to sort of start digesting it now. And I believe the book you mentioned is The Immortal Life of Henrietta Lacks, if my title memory is right. Awesome. Well, thank you so much. Is there anything else that you want to tell us about? before we sort of sign off?
I just wanted to share that as somebody, I wish that I had the skills that you have now coming out of this course when I was undergrad. Because when I graduated, I didn't have the opportunity to really learn these techniques as well as you have. Your immunocytochemistry, your staining skills, your microscopy skills, far and above what I was able to do at your stage and what I think most people are able to do. It was really beautiful work, and that was really fun to see. It was so much fun to see you guys grow throughout the semester and try new things and fail, but also really succeed, and that was really awesome. Yeah.
Thank you for all your help this semester. You're the best GTA.
Awesome. Well, Ariana, thank you so much. I really appreciate you coming and talking to us today.
Thank you. It was so nice meeting you.
I'm joined by Gus Peters. Gus, would you mind telling us a little bit about yourself?
Yeah, my name is Gus Peters. I'm a junior MCDB major.
What do you plan to do after you graduate?
Yeah, after I graduate, hopefully I'll go into med school. I'm taking the MCAT this summer,
Nice.
so
Good luck.
fingers crossed for that. Yeah.
Cool. And then is there a specific kind of doctor you want to be?
I want to go into anesthesiology, hopefully.
Nice. So would you tell us about your research project and why you picked that treatment?
Yeah, so me and my lab partner, we wanted to look at how phone lighting can impact cell health. Specifically, we wanted to look at actin filaments in the cytoskeleton. So we really wanted to look at it just because we're students, we're on our phones a lot. A lot of other students are also on technology of a ton of other forms. And we just wanted to see how that impacts our health and if we could maybe cause better health outcomes.
Yeah. So this would be specifically for blue light, correct?
Yeah, so we wanted to look at blue lighting in phones, but we also wanted to look at warm lighting. So we tested with both like a normal phone and then also a phone that we turned night mode on to see if there would be any differences there.
So with the two different light settings, what did you find?
Yeah, so it was really weird. We looked at the two different light settings and we expected a lot of damage in these blue light exposed cells because, you know, blue light carries more energy. We thought maybe it would depolymerize there. And what we actually found was that the warm light exposed phones had more damage. Like they showed odd nucleus shapes. They had very depolymerized actin. And the normally phone exposed cells actually showed closer to our controls. They were relatively stable.
That's fascinating. So just kind of for a mental picture, were you just like holding your phone up to the cells?
So we took our phones. It was my phone and then my lab partner's phone. And what we did is we put them on max brightness. We just put a white screen on there. We made sure all the notifications were turned off so we wouldn't get any interruptions while we were testing. And then we just put that on top of the cell plates and then covered all the cell plates with a box so it was only the phone lighting and just left them like that for 30 minutes.
How did you decide 30 minutes?
We thought that would be good enough to just give a result.
What more can you tell us about your findings?
So our findings, once again, they were like very odd, not what we expected. So when we exposed the cells to what I'm just going to call a normal phone, regular phone lighting, we saw that actin depolymerize a little bit. The nucleus was slightly bean-shaped, which shows that maybe it wasn't completing mitosis correctly. But for the most part, the changes were very minimal. And then when we looked at the cells exposed to warm light, there were very odd things happening. So there were a lot of little lobes coming off of the nucleus, which maybe shows damage there. Many of the nuclei were that bean shapes. They weren't dividing properly. We saw quite a few less cells. And then that actin was just kind of wholly demolished. It was very clustered around the nucleus, but every other filament just kind of wasn't there.
This is not good news for the blue light glasses people.
It's not good news for the blue light glasses.
So they don't work is what I'm learning, right?
Yeah, we were hoping to test blue light glasses, but we didn't get the opportunity to do that. So maybe they're different.
Yeah.
This is interesting. This is not what I would have expected either because of the reasons you said.
Yeah.
But I think that you guys did a really good job designing your controls. So you had like all the light because of the warm light mode. What that does, right, correct me if I'm wrong, it kind of screens out that blue light coming through, right?
Yeah, we couldn't find like exactly what it does, but it makes like the screen more yellow. We're kind of assuming the wavelengths are getting shifted towards that red end. They keep their trade secrets close. We couldn't figure out what they really meant by all of it.
I'm kind of wondering, because I think about like in the beauty world, there are like red light masks, and they're talked up as things that are like anti-aging, but it sounds like red light is more destructive than the blue, if I'm recalling correctly, or at least from what you kind of seen.
It would be worth testing that. I know UV damage can cause a lot of issues with the skin. I don't know what a red light mask would really do for you.
I had always kind of wondered.
Well,
we got Evan. Yeah.
This year we actually had some students test a red light therapy device on their cells, and they saw that the red light treated group actually their actin filaments were less depolymerized and like stronger and healthier than their group that was treated with just normal ambient white light. I'm not sure what that means in terms of like whether that's a good thing or a bad thing, but it's almost kind of different from what you saw, where you saw the warm light group was more depolymerized. The red light therapy group, which is that's a totally different like kind of treatment because they're getting like a dose of like just red light, where the phone is probably emitting a broader spectrum at a different kind of intensity. So we had a number of groups test light-based questions, which is very interesting. And yeah, there's a lot of very topical light-based things that are really prevalent right now in society, whether it's because of technology or it's because of research like this that has led to questions like new, really weird, innovative ways to approach things like cosmetics. You guys came up with really interesting questions. And so, it's actually made me kind of really think about things in my own life because I have my phone like 3 inches from my face a lot.
Yeah, I went to the eye doctor right before the results of this experiment we got him, and they were asking me there if I wanted blue light filters put on my lenses. And after this, I'm like, I don't know if I want that anymore.
Interesting.
Yeah.
So they actually make... Oh, interesting.
Mm-hmm.
Mm.
Huh. Yeah, that's fascinating to know. And now I'm like thinking. So much more I'm aware about. All right. And then in regards to the experiment, is there anything that we did not talk about that you think we should touch on?
I don't think so.
Okay, cool. Yeah. And so what did you enjoy most about the experiment and like working in the lab?
I learned a lot of cool lab techniques. I've not gotten to do many of the things that we've done in this class before. So we got to like count cells, we got to stain them, we had experience culturing stuff, using all the centrifuges. It just felt honestly very professional. It felt like I was working in like a really focused lab, and I have not had that experience before at KU, mostly because I haven't taken other biology lab courses. I'm sure they're all great, but this one was very fun.
Yeah. I mean, I'm glad that you had an opportunity to get a really hands-on experience because I'm sure it changed kind of how you're viewing, these college courses and possibly even what you're planning like to get onto and do later in life.
Yeah. It was also nice just being able to design like your own experiment, to just take those skills that you learn and then really look at something that you want to look at. Like it made the class feel like it built somewhere, and I really enjoyed that.
How do you think all of the experiments you did over the course of the semester are going to benefit you in the future?
Yeah, I'm taking more lab classes at KU. Hopefully this will be a lot better for me now that I know how to run more experiments. I'll be working at KU over the summer in a lab, and hopefully this will show me how to like stain and stuff if I ever want to test those sorts of questions.
Yeah, I think you're really prepared to work in a lab and to progress with your research here because your staining was beautiful. Your not just your immunocytochemistry, but also your Western blot work and your cell culture work was all fantastic. And I think you've really grown a lot as a scientist. And like you're saying, I think you became really pretty professional over the course of the semester at what you were doing. So that was really fulfilling for me to see.
What about the lab did you enjoy most?
I enjoyed just spending time in there. I've been in a lot of chem labs as opposed to biology labs, and I think chem labs feel like a grind, which is maybe just because I've always taken them at 2 p.m. and gotten out at 6 when it's dark out. But I liked just being in this lab because it very much felt like the time that was passing was still productive, and like waiting on experiments actually meant something instead of sitting in chem lab waiting for something to heat up for an hour and a half, which is not a slight against chem lab. I'm taking 2 chem labs next semester. But I just like this different experience of being able to run lab work.
Thank you you were a really fun part of class. And actually, you were a really deep thinker. You thought very deeply about the questions you asked with your independent experiment. And you were also just a really creative thinker. So that's something I really enjoyed having about you in class.
Thank you. Yeah, you were a really good TA. I enjoyed the class. I was a little horrified looking at your cell staining when it had like 15 colors on it, and I was watching what it takes to make like 2 or 3. But it was a really fun class.
Thank you so much for joining us today. Really appreciate you taking the time.
Yeah, thank you.
We're now joined by Toby Brown. Would you mind telling us a little bit about yourself?
Yeah, sure. So I'm a molecular, cellular, and developmental biology major with a minor in sociology here at the University of Kansas. And I'm involved in biomedical and public health research. And as a non-traditional student, I'm also actively engaged in parenting.
As a non-trad student, do you have any advice that you would like to give people on sort of that track?
Absolutely. I'm glad you asked. Thank you for giving the opportunity. Yeah. I would highly encourage all non-traditional students to know that you are at the right spot. You are able to access the resources and the knowledge you need to meet the goals that you have here at the University of Kansas. So never feel like you are incapable of pursuing those goals. It may be a little more, may require a little more, strategy, but you can do it. Trust me, you can do it.
Mm-hmm. And then if I remember right, I believe you're graduating this year.
Yes,
Congratulations. That's amazing news.
Thank you so much. I'm so excited.
So what are your thoughts for the next step?
So my next step is I've just earned entrance into completing an internship with the thesis program. So I'll be doing some indigenous research in Oklahoma over the summer.
Wow,
that's amazing.
Awesome.
Really excited.
Congrats. Would you mind telling us about your research project and why you picked that particular treatment?
Yeah, absolutely. My lab partner and I researched how different electrolyte concentrations affect HeLa cells by treating them with varying dilutions of a commercial electrolyte solution. And this allowed us to simulate acute changes in electrolyte conditions. So we chose this treatment to try to better understand how electrolyte solutions influence cell structure beyond just their typical role in human hydration.
What did you end up seeing?
Well, we found that both low and high electrolyte concentrations altered cell growth and morphology compared to our control group. So we tested this using GFP and DAPI fluorescence microscopy and staining to observe the actin, cytoskeleton, and cell nuclei, which permitted us to clearly detect structural differences.
Yeah, so I'm a bit of a visual learner. So, can you describe, what those structural differences were for us?
Absolutely. So, thankfully, because we had guidance from our GTA, Evan, and, of course, Dr. Houston, we were able to know that sometimes when you're looking at the morphology of a cell, you will notice that sometimes the outer boundaries, specifically the cytoskeleton, will be stretched or maybe shrunken or maybe expanded. So usually a cell is they all have kind of a pretty much variable similarity in their structure. But when we were exposing these to an extracellular concentration of electrolytes, we noticed that there was shrinking in these particular cells, which was right along the lines of what we had predicted. So it was really cool to be able to see that, in fact, that these concentrations of electrolytes did, in fact, shrink the cell, which was right along lines with our prediction.
Why were you expecting to see cell shrinkage?
Yeah, thanks for asking. So we initially, to be really honest, we were predicting that actually there would be expansion. So we actually had thought, there's going to be this growth. And clearly, when we started thinking about things about, like, when you add salt to something, it typically is going to shrink. We started realizing, maybe we ought to realign our thoughts here. So from the layman perspective, which I can identify very easily, usually, we did have to kind of analyze and do some critical thinking about the concepts involved with this shrinking, the idea of hypertonicity, which is like the shrinking and expanding of cells based on the concentration of solutes. So the idea was just that we were trying to think about the fact that typically we all hydrate with electrolytes, but what are they doing on a molecular and cellular basis? So that's kind of what we were looking to do here.
And that's a really interesting point. I thought it was so cool how you guys picked your project. I actually really liked it because we're not allowed to name brands here because we want to avoid like a lawsuit. And I don't know if this brand has like a catchphrase. Electrolytes. Often, whether they're blue or they're yellow, that's typically the kind of flavorings that they're called. Mm-hmm. It's very popular that we drink it all the time. Actually, yours, the product that you used was colorless, wasn't it?
That's right, Evan. Thankfully, you and Dr. Houston had to try to encourage my partner and I to use critical analysis with thought for also the potential confounding variables that we wanted to try to create do some research that didn't have alternative reasons that the cells could be changing with regard to their morphology and structure. So thankfully, because of your guidance, we were able to find a commercial electrolyte solution that was in fact colorless and had no additives. So it was simply electrolyte water. Mm. So it was ideally something you'd want to use with regard to like an experiment because there was less opportunity for those confounding variables to be what's impacting what we saw in our results.
And I was actually really, really blown away by the results you did see. I kind of thought, I don't know why I thought this, but I kind of thought like maybe because your exposure wasn't like super long. And I was kind of thinking that because we drink a lot of this, that it wouldn't be that disruptive. But it was. The state of Florida is not going to be happy about this, but it, it was so interesting. I mean, I'm not necessarily thinking it's doing that to our bodies, but like even just to see how the change in electrolyte concentration affected the cells was really interesting.
And Evan, that's exactly what made this so cool, was that we all know that we need electrolytes when we're working out. We're sweating, we're losing our solutes in the form of sweat. But when we're looking at this from that molecular and cellular level, it's like, this is what happens, but how does hydration work if we need electrolytes? So it was really cool to see it at the molecular and cellular level. Like you said, that's really cool.
In a way, I sort of feel like it just reiterates how important paying attention to serving size is. Because I know I've had some of like the powder stuff when I was sweating a ton, doing extreme hiking, and there's very much that you do not want to go over the limit per day. So, yeah.
Maria, with that very, what you just said, that's exactly what the future plans are with this research. There's research right now that's looking at trying to personalize the medicine and the use of electrolytes based on the person's, variable characteristics like height, weight, because they're saying that, just like you said, that there is the need to consider that, for example, I'm a different weight and body mass compared to you and Evan, so my need for electrolyte amount is more than likely going to be variable with, in comparison to what you all may need. So yes, these are all future directions and directives that are being actively researched as we do this podcast.
Mm-hmm. Yeah, and I think it's a good point because the classic phrase, "The dose makes the poison." Mm-hmm. The dose also makes the medicine. So that's a really interesting point that you bring up.
Yeah,
Indeed.
And honestly, glad to know that they're like making it more specific to people, because I think within the last few years it came out, or at least I became aware that like they didn't start testing drugs on females until I think it was the year I was born, 1993. And so like, just sort of think about that. It's only been 30-esque years of having everyone being tested. So like hearing about sort of micro testing is, that's thrilling.
Yeah, that is a really important expansion that's going on in the field of biomedical science. And I appreciate, it sounds like you're also going to be kind of doing that with your own career, is looking at subclassifying how different things affect different groups of people. I think that's really, really important. Just to like your point, Maria, about it not being tested in women, I think, and this is a complicated issue, but one group of people that isn't tested very often for drugs is pregnant people. And there's obviously reasons why people would be afraid to test that, but it also means that we just don't know. So there's all kinds of, and there's beyond just, biological sex and all that kind of stuff. There are so many groups of people that are just underrepresented in these studies. And I'm really excited for you, Toby, to in your career. I hope that's something that you can always keep with you and a focus that you can keep with your work.
Yeah.
Thank you, Evan. I appreciate your comment, and I do. I definitely agree with you that the need to, have greater data on less represented populations so that we can, make better opportunities for personalized medicine and other, biomedical related needs regarding people and their health.
Mm-hmm. Yeah, super important. So I'm just glad that we're seeing a change in, things will get better, right? It's what we hope. What did you enjoy most about your experiment?
I, as Evan had shared earlier, I found it most exciting to be designing this independent freestyle experiment and to be able to then actually make visual these results, how it all came together. It made me feel like an actual practicing scientist, and I cannot say that I've ever felt like that, and my confidence has increased because I was able to get that hands-on experience and put forth action toward things I'd been reading about for the last 4 years. I got to actually participate in them. And so it's really, really amazing to be able to say that I now have the confidence, now feeling more like a scientist these days.
How do you think all of the projects and experiments that you did in this class and the lab, how do you think that's going to benefit you moving forward?
This experience helped me build essential lab skills and strengthen my critical thinking and data analysis abilities, which will definitely be valuable in my future regarding biomedical science and medicine.
Yeah, I just want to say, you actually, it was really amazing. You guys got amazing results. Your immunocytochemistry was beautiful. Your Western blotting was beautiful. You guys also did a great job in the cell culture hood. You guys did a great job all semester. You grew immensely. And I would say, you and all of your classmates are all incredibly qualified to do these things in the real world for real scientists, as real scientists, because that's what you guys did. You guys did real science. You tested a real scientific question by real scientific means. It was you. It's been a joy to have you in the course. I'm really glad that you asked such an interesting question, because I think that is really what it's all about. And I think that, kind of to what you were saying, Maria, figuring out if you really enjoy something is critical. For all the students out there, really just make sure that you're doing something because it's something that you enjoyed doing and that you can see yourself getting excited about going to work every day, because you'll have to go to work every day.
Mm-hmm. Yeah, enjoying your job makes the daily grind so much better. And KU's been great to me, but the job I had before wasn't. And so it's just remember, you're never stuck where you are. You can always get better. You can always change what you're doing. But it helps knowing what you like.
That's right.
Yeah. And I also want to say, there is no linear path. I think a lot of students have this idea that there's like this way that you're supposed to progress, and I can tell you that wasn't the case for me. That's not the case for really anyone, I don't think. And it does not matter so much as you're doing what you feel fulfilled doing and that you're making progress towards your goal. So if things change, don't get discouraged, keep an open mind. And then also taking courses like this really do help you figure out what you enjoy physically doing, because you'll actually physically do these things, not just read about them in a textbook, you will physically do them. And I've had students in this course think that they wanted to go in a particular career direction, and they actually fell in love with doing research. And they, from there, joined a lab and actually are pursuing more of a research-oriented career. So getting that hands-on experience with whatever it is, I think is critical.
Yeah, honestly, great advice. What part of the lab did you enjoy most?
I really enjoyed working alongside my colleagues. They're incredible and bright, and they provided both academic and insight, and they provided a sense of community toward me. So that made my experience even more rewarding.
Yeah, Toby, you elevated the class. So, like, you are a people person. You are fantastic with the people, and there were days that I came in that I was kind of having a crappy day, and you made my day better, and you made it, like, because very much people, science is a people thing. It's very, much based off of our own cultures and even just the vibes we're putting out. So if I'm not putting out good vibes, the class isn't going to have as good of a time. It's not going to go as well. And there's been a number of times that you actually made the class go better because you brought your own, interpersonal skills into how you interacted not just with your students, but with me. And that was just so much fun. You were a blast to have in class, yeah.
Thank you, Evan. And that all goes back to exactly what you said about this not being a linear path. Because my path is not linear, I think that helps me to be so thankful for being in the position I am right now, like taking cell biology lab. That was something that I am so incredibly thankful that I got to participate in as a student.
Oh, I'm glad to hear that, because I was actually very inspired by you when I learned about your path. And the work you put in is amazing. You put in so much work, and you have to go when it comes, sometimes, nonlinear paths are just fine, but sometimes they do mean that you have so much more to think about and so much more to do, and that's just been really, really inspirational for me. Your ability to do that is amazing.
Thanks, Evan.
Toby, thank you so much for joining us today. It was amazing to hear about your research, about where you plan to go in life, and yeah, it's just been great talking with you.
Thank you, Maria. I'm so glad I got to finally meet the GOAT in person.
Thank you. I want to thank all of our student researchers for joining me today. I hope hearing about their research inspired you to learn more about everything we talked about. Thank you again for listening to Interview with a Biologist. If you'd like to learn more about KU biology degrees or the classes offered here, please check out biology.ku.edu.